Yeah. Some scientists think the discovery of the structure of DNA is the greatest discovery in the history of 20th century biology, if not science. So it's super important in the world we examine today, in the world we live in. The advent of DNA sequencing, which was only possible because we were able to determine the structure of DNA, has led to things like solving 50-year-old cases, murder cases, rape cases. Many of these have been solved through DNA sequencing technology. So very important.
Should you cut your finger on Band-Aid?
No. Does anybody have a small band? Yeah.
Wait, I actually
I don't
might.
know. I don't want to get something funny.
Unless I took it out. I just had one. Oh, I do have one.
It's not small.
I have a small
You have a small
one like this. It's from
Just the pen.
From Greece. It is.
What do we gotta say? Oppa!
I literally cut my feet and then I had to buy someone a Band-Aid and I left it in the school. Forgot. Some Greece Band-Aid. Souvenir. I love those.
Yeah, so today's lecture is how many slides? It's 53 actually. I don't think I'm going to get through the full 53 today. Hey, Grant. Your next quiz will be on chapter 7. It's not going to be this week. I'll announce it at least a class before, or something like that before. Probably a class before. But it'll at least be on the announcement on campus. But your next quiz will be on chapter 7. Okay. Yeah, got it, my thing downtown? Yeah. Oh, my third. Discuss DNA in Unit 1. DNA is the molecule that makes up your genes. Your genes are DNA. Okay? Your genes, your genetic information, that determines what type of organism you are. There's a reason when, say, two humans mate, they give birth to a human. When two whales mate, they give birth to a whale. The reason that is because they pass on their DNA to their offspring. So their offspring looks like them. What do you guys think? What came first, the chicken or the egg? Who says the chicken? Raise your hand. Who says that? It's the egg. Okay, because mutations that build up in the gametes, the cells that make up the future offspring, they carry the mutations that give birth to a new species. That means the first chicken was produced in an egg that was laid by an organism that wasn't a chicken. So it was a different species from its parents. So DNA, what is DNA? Who knows, what is the classical name of the structure of DNA?
I can't say it. I know what it is.
It's a double helix. The double helix. So it's two helices wrapping around one another. Okay? Now, the units that make up these helices are called nucleotides. A nucleotide contains three components. Who knows, what are those three components?
A sugar and three phosphate.
Phosphate, a sugar, and what else? A nitrogenous base. Nitrogenous base just means that it's a base that contains nitrogen. Okay? So nucleotides are the units that make up the double helix. They consist of three components: a sugar, a phosphate, and a nitrogenous base. Okay, now in DNA, what are the four nitrogenous bases? Adenine, that's one adenine, what's the other? Thymine, cytosine, guanine. ACTG. Now, wherever there's an A in one helix, what is there on the other helix? T. Is that true in reverse? If there's a T, is there an A?
Yes.
Yes. Okay, now if there's a C in one helix, what is there on the other one? G. And is it true in reverse? Yes. So wherever there's an A in one helix, there's a T in the other helix, and vice versa. And wherever there's a C in one helix, there's a G in the other helix, and vice versa. Okay. Right. So, the incredible diversity of life is determined by the information within DNA. It's composed of four nucleotides: adenine, thymine, cytosine, and guanine. And this can code for an enormous amount of information. So, three nucleotides, one after the other, to determine one amino acid. Okay? So, how many types of bases are there? Four. So, if three bases in a row to determine the type of amino acid, how many possible combinations can be made? If you have four different in the first one, you have four possibilities for the second one, and four possibilities for the third one. Who remembers from algebra? Four to the third. You have 4 times 4 times 4. So what is that? 64 possible combinations can be made. Okay, now, how many amino acids are there? 20. So you have 64 combinations, but only 20 amino acids. What do you think that tells you about the code? It's said to be degenerate. In other words, multiple codons can code for the same amino acid. A codon is just 3 successive bases. Okay? It's called a triplet codon. 3 bases, and different codes can encode the same amino acid. Different sequences can encode the same amino acid. Now, there's a codon called the start codon. That tells the ribosome, start making the protein at this sequence. Does anybody know what that start codon is? Yes, AUG. AUG. Whenever that code is present in a gene, it tells the ribosome, start translating here. Start creating the protein here. AUG. And does anybody know, what is the amino acid that is encoded by the start codon? It's methionine, the amino acid methionine. Methionine. Methionine.
How do you spell it again?
Methionine.
So that's the amino acid.
Methionine. That is the amino acid encoded by the start codon. So, if you have a protein, you know the first amino acid is methionine, unless the protein is altered in some way to remove that methionine. But the first amino acid that's translated is methionine. Okay. So that's our start codon. There are 3 stop codons. These tell the ribosome the protein has been created. You can stop translating the gene into protein. Those three stop codons are UAA, UGA. Did I say UAG? Yeah, I said UAG and UGA. UAA, UAG, and UGA. So we have the start codon, AUG, and the stop codons, UAA, UAG, UGA.
Wait.
What?
What kind is this?
AUG.
And then those three all the time.
Yes, any one of those three are stop codons. What's that?
What was it again? Stop
The
codons.
stop codons?
Stop codons.
Stop codons.
Wait, this is the, do you need to stop? Is this the stop? I don't know.
Does anybody know who was the person who came up with the theory of the gene, or whose work led to the idea of the gene? He was an Austrian monk in the 1860s. Who knows? Gregor Mendel. Yes, he worked with plants, and he came up with the concept of the particulate theory of inheritance. Before the particulate theory of inheritance, there was an idea that offspring tend to have traits that are intermediate between those of their parents. In other words, if you have a tall parent and a short parent, then the offspring have a height that's medium. There was this blending. But Mendel introduced the idea of the particulate theory of inheritance. In other words, parents pass on specific units to their offspring that determine what traits they'll have. Today we call these particulate units genes. You guys remember in Unit 1 we talked about Frederick Griffith? You guys remember what I said about him? Griffith was working on pneumococcus, a type of bacteria. He was trying to develop a vaccine against pneumonia. He was working in the early 1920s. Why do you think somebody would be working on a vaccine for pneumonia in the early 1920s? What happened just in a few years prior, 1919, what happened? Not the plague?
Influenza.
Spanish flu, right? The Spanish flu. So Griffith was working on a vaccine for pneumonia. There were two strains of bacteria he was working with, these pneumococcus. One was called an S strain. It looked shiny and smooth. Okay, today we know it has a capsule. Interestingly, we're doing capsule staining today. The other strain was called the R strain. It appeared rough. Okay. Now, the upshot is that the S strain is deadly. It's virulent. The R strain is harmless. It's not virulent. So if you take the S strain and you inject it into mice, what happens to the mice? They die. What if you inject R strain bacteria into mice? They live. Okay? Now Griffith did something interesting. He took S strain bacteria, he killed them through heat, and then he mixed them with living R strain bacteria. And then he injected that mixture into a mouse. What do you guys think happened? They died. Not only did they die, but live S strain bacteria were recovered from the mice. Okay, so how did that happen? There was this idea that whatever transformed the R strain bacteria into S strain bacteria was called a transforming principle. This word principle is like a medieval philosophical word, but it's a substance that can transform a trait in bacteria from one thing to another. Okay? Today, we recognize that it is the genetic material. It is genes. So Griffith began work. He wanted to purify this transforming substance so we could get down to the idea of what are genes, what molecules make them up. Unfortunately, Griffith was killed in 1941, during World War II by a German bomber. He was in Britain during the London Blitz, and a bomber killed him. So he was unable to do so. But another group of researchers, led by Oswald Avery and his colleagues, they set about this work. They worked for 16 years. They did 16 years of research before publishing their results in 1944. And basically, they took this transforming substance, they exposed it to material that destroys protein, material that destroys RNA, and material that destroys DNA. And when they exposed it to material that destroys protein, it had no impact on the transforming substance. When they exposed it to material that destroys RNA, it had no impact on the transforming substance. When they exposed it to material that destroys DNA, it was unable to, do you have a question? Yeah. It was unable to, it did have an impact. So the animal couldn't, or the model organism they were working with couldn't be transformed. So Avery and his colleagues published their results in 1944. And the scientific community, basically, they responded with skepticism. You probably had an impurity in your sample, or you did something that skewed the results, so they didn't believe them, basically. Even though Avery's results should have sealed the argument, for sure. And in fact, Avery is often considered the person most deserving of a Nobel Prize who never won one. So we would have to wait until 1952, when two researchers, Hershey and Chase, they did their experiments on viruses, which effectively determined that DNA is a genetic material. And the following year, in 1953, two researchers, Francis Crick, a Britishman, who was by training a physicist, and James Watson, an American, they looked at X-ray images of the molecule of DNA, and they were able to determine that it is a double helix, and they gave us a structure of DNA that we have today. So from 1865 till 1953, that's how long it took us to learn that DNA is a genetic material, about a century, 100 years. Okay, for a question that to you guys is so commonplace, it took a century to learn that. Okay, so science, especially when it first begins, moves very slow. Today we're in the age of AI. Discoveries are being made that would have took previous researchers decades to make. We can uncover them in months. All cells contain DNA. All cells contain double-stranded DNA. Viruses don't necessarily have double-stranded DNA. They can have DNA or RNA, and it can be either double-stranded or single-stranded. So all cells have DNA. Viruses have RNA, it can be single-stranded or double-stranded. So in order for a cell to multiply, one of the things it must do is replicate its DNA. A gene can also be expressed. In other words, you take a gene and you use the information in that gene to make a protein. Before you can do that, you need to make a complementary RNA sequence of your gene that's in DNA. So you have your DNA, you make a complementary RNA sequence, and then you take that RNA sequence outside of or to a ribosome, and there you translate the RNA sequence into a protein. So the process of converting DNA or making RNA from DNA is called transcription. Making RNA from DNA is called transcription. Making a protein from RNA, that's called translation. Making RNA from DNA is called transcription. Making protein from RNA is called translation. So information flows from DNA to RNA to protein. This is sometimes called the dogma of molecular biology, that DNA becomes RNA becomes protein. Well, not long after Watson and Crick determined the structure of DNA, there was an enzyme discovered by an individual named David Baltimore. He recently passed away. But the enzyme is called reverse transcriptase. And it can convert RNA into DNA. Okay? So, within less than a decade, some people said the dogma of molecular biology had been broken. Well, if you look at what Francis Crick actually wrote, he didn't write that DNA goes to RNA goes to protein. He said nucleic acids become amino acids. Information flows from nucleic acids to amino acids. He didn't actually say DNA to RNA to protein.
What was the name of the enzyme again?
Reverse transcriptase. That's an important enzyme. Does anybody know what microbe expresses reverse transcriptase? HIV. HIV, what's that? HIV expresses reverse transcriptase. These viruses are called retroviruses. So the two strands of DNA, they are complementary. In other words, we've already mentioned that wherever there's an A in one, there's a T in the other, C on one, G on the other, and vice versa. That's called, that's referred to as being complementary. They complement one another. Another feature of the two strands of DNA is that they're anti-parallel. So one end of a strand is a 5' end, it's called a 5' end. The other end of the strand is called the 3' end. Well, if you look at the two strands of DNA, the 5' end of one strand is next to the 3' end of the other strand, and where the 3' end of the first strand I mentioned, next to that is a 5' end. So they're called anti-parallel and complementary. They're held together through hydrogen bonds. Okay? So wherever there's an AT in DNA, that is an adenine bonded to a thymine, you have two hydrogen bonds holding them together. Wherever you have a G and a C somewhere in DNA, there are three hydrogen bonds holding them together. So AT2, GC3. That's important because when you have organisms that live at very high temperatures, they tend to have many more GC bonds than they do AT bonds.
Because of the number of hydrogen bonds that allows for that.
Oh, well, it makes it more difficult to break apart the helix. Okay, because there's more energy holding it together with 3 bonds rather than 2. So separating the two strands of a DNA molecule, that's called melting or denaturing. This can be achieved by heat. So DNA replication. Who knows, what is the name of the enzyme that unwinds the double helix? It melts or unwinds the two strands of, separates the two strands of the helix. Unzips the two strands. It's called helicase. There's a famous joke about helicase. Let's see if we can look it up.
So is that the same thing as melting or denaturing as helicase?
Oh, yeah. That was very good. The famous helicase joke. I was in AP Bio back in 2010. Okay. So once the two strands are unzipped, you need to stabilize the two strands. Okay? This is achieved through what are called single strand binding proteins. So the helix is unzipped, and then single stranded binding proteins bind to each strand. Okay? To stabilize them.
What are those called?
Single strand binding proteins, or single stranded binding proteins.
And they bind to each one of the, is it helices you said?
Each one of the unzipped strands. Not helixes, strands. Yes. Now, in order for you to, so each strand is used to make a complementary strand. Okay, so you need an enzyme called DNA polymerase to do that. It will extend, it will produce, it will make a complementary strand of the helix. However, DNA polymerase cannot create a new strand from scratch. It needs it already needs a nucleotide sequence already present in order to create, in order to extend that, what's called a primer. So these primers are provided by an enzyme called primase. Okay. And what do you think these primers are made of? Are they made of DNA or RNA? RNA. So primase is an RNA polymerase. Okay. An important point. As you continue to extend these the complementary strands, you have to continually unwind the helix. And that creates torsion in the helix. In order to relieve that torsion, you need the help of a group of enzymes called topoisomerases. Okay, these break the helix, reform it, break the helix, reform it in order to relieve tension.
What's it called again?
Topoisomerases. A topoisomerase that's present in all or nearly all bacteria is called DNA gyrase. It's a major topoisomerase.
How do you spell topo?
Topoisomerase, Topoisomerase. So DNA gyrase is a major topoisomerase. This is important. By the way, it's been recently discovered that DNA gyrase is also present in some archaea. Okay? And it's not as common, but there are some DNA gyrases present in archaea. However, they're almost ubiquitously present in bacteria. And this is important because certain antibiotics specifically target DNA gyrase, which is not present in humans or higher eukaryotes. Okay? Have you guys heard of the antibiotic Cipro? Cipro is a type of antibiotic known as fluoroquinolone. Okay? Cipro targets DNA gyrase. This is important. When we develop or discover antibiotics, we want them to target something that's unique to bacteria and is not present in humans. Because if it's present in humans, it will also target our own cells and we could be killed by it. So we need to target things that are unique to the microbe we're trying to kill or prevent from replicating or dividing. So DNA gyrase is targeted by certain antibiotics. One of them is Cipro. Cipro, by the way, is often used for infections like anthrax. So the double helix, you have a sugar, you have a phosphate, a sugar, a phosphate, a sugar, phosphate, sugar, phosphate, sugar. On the other strand, you have a sugar, phosphate, sugar, phosphate, sugar, phosphate. This is what we mean by anti-parallel. the bond that connects a phosphate and the sugar is called a 3-5 bond. Because this is the first carbon, 1, 2, 3. So, at this third carbon, we have a bond between phosphate. Well, where is this phosphate bonded to on the other sugar? 1, 2, 3, 4, 5. Okay, so it's a 3-5 bond, 3-5 linkage. By the way, the sugar in DNA is what? What is the sugar called in DNA? Deoxyribose. What is the sugar called in RNA? Ribose. Okay, so we've already mentioned one difference between DNA and RNA. DNA has the sugar deoxyribose, RNA has the sugar ribose. Another point. In DNA, we mentioned that there's ATCG. Which one of those is not present in RNA? T. T is not present in RNA. What is present in RNA? Uracil. And that takes the place of the T in RNA. In DNA. Okay? So it's the same thing. In RNA, instead of having an AT linkage, you have an AU linkage. Wherever there's an A, there's a U, and vice versa. Another difference between DNA and RNA. DNA tends to be double-stranded. What does RNA tend to be? Single-stranded. Okay? Which one is more stable, RNA or DNA? DNA is more stable. The reason for that, let me see. I'll show you why. I'll show you what I mean. Here is an image of DNA. You see this second carbon? What is it bonded to here? A hydrogen. This is deoxyribose. In ribose, you see the second carbon, what is it bonded to? A hydroxyl, an OH. And as some of you guys will learn, if you take organic chemistry, that hydroxyl group is quite reactive. Okay? That hydrogen in deoxyribose is not very reactive. The hydroxyl in ribose is very reactive. And that's what makes ribose RNA so much less stable than DNA.
Sorry, could you repeat that?
Yes, the OH in ribose, this OH makes this ribose much less stable than this deoxyribose. The reason that's the case is that this OH is much more reactive than this H. You can look at it here too. There's another image. The blue is the hydrogen, the pink is the hydroxyl. Okay? And if you guys remember the RNA world hypothesis I told you about in Unit 1, where all living systems were originally composed of RNA, if RNA had remained the genetic material, life couldn't have evolved to the extent that it has. Simply because RNA is far too unstable to support living organisms today. It would just accumulate massive amounts of mutations. And that in turn would, it would have prevented life as we know it from coming to existence. Okay. DNA replication is usually bidirectional. In other words, because DNA is a helix, as the complementary strand is being synthesized in one direction, on the other direction it is also being synthesized on both strands. So if you're going to the left, one of the strands is being synthesized complementary, the other strand is also being synthesized complementary. As you go to the right, the same thing. You have one strand that's being synthesized that's complementary to one, and another strand that's being synthesized that's complementary to the other strand. Okay, so both strands are being synthesized, complementary strands are being synthesized of both in both directions. Does that make sense? Yeah.
Okay, so I'm trying to put it together in my head. So are you saying like the double helix is like breaking and like one side is having a complementary side to that and then the other side like what to like multiply or?
Give me 1 second. So you guys see this double helix, these two helices wrapped around one another, or these strands wrapped around one another? Let's say you started here, okay? And you wanted to you wanted to do DNA replication. All right, let's start here. If you go in this direction, both strands, you're making complementary to this strand and this strand. So this strand, the blue, and this strand, the red, you're making complementary strands to both. Make sense? So let's just start in the middle, just a random place. You guys see I'm in the middle here? Okay. You have a blue strand and a red strand. So you're gonna make a complementary strand to the red one and the blue one. You're making complementary strands to both strands. That's in one direction. On the other side, the same thing is going on. You're making a complementary to a red and a complementary to blue. That's what we mean by bidirectional. Because you have to start replicating DNA somewhere. And in order to do that, you have to unwind the helix and you have to synthesize DNA in this direction and this direction. So it's bidirectional.
So is it 4 complementary strands being made or just 2?
Remember that on one side, they're the same
It's
strand.
going like this?
Yeah.
Kinda, okay.
Yeah, it's going like this. It's bidirectional. Okay?
And that's to make proteins or?
That's to make a complementary strand of DNA. So wherever there's an A in one, a T in the other. So you're replicating DNA. That's not to make proteins.
Okay, so does that like happen naturally or like what is it being copied for? For
Yeah,
cell to
in order for the cell to divide,
Oh, okay.
you need to replicate the DNA.
Oh, I see, okay.
Let's see if we have a,
Doesn't matter where you start that process at?
for our purposes right now, it doesn't matter. You don't need to know that. However, there can be certain areas where it starts. Let's see.
introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where. Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when. When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember,
I just want to show you guys the bidirectional.
Yeah, personally.
This is good.
I knew what I passed.
So these are the two strands of the helix.
DNA replication begins at specific chromosomal sites called replication origins. At the origin, the DNA duplex is melted by specialized proteins, resulting in a replication bubble. DNA synthesis starts at the origin and proceeds bidirectionally. Two replication forks move outward in opposite directions. At each growing replication fork, 2 new DNA strands are made. The leading strand is synthesized continuously from a single RNA primer. It grows in the 5' to 3' direction, the direction of replication fork movement. The lagging strand is copied discontinuously from multiple primers, resulting in a succession of Okazaki fragments. Although the direction of DNA synthesis is opposite replication fork movement, the overall direction of lagging strand growth is toward the replication fork. As replication proceeds, the RNA primer of the first Okazaki fragment is replaced by DNA. Then the lagging strand fragments are ligated together.
Do you guys want me to post this video? You guys want it? Post it. I'll make a note. Alright. YouTube helps, guys. When I was in dental school, some of the other students were, so, by the end of DNA replication, you're gonna have 2 helices of DNA. The original DNA and the new DNA. The key point, in each DNA, one of the strands is gonna be from the old strand, and one of the strands is gonna be from the new strand. And the same thing in the other DNA molecule. Okay, that's why it's called semi-conservative. Each DNA molecule consists of 1 old strand and 1 new strand. Okay, now that might seem obvious, but you could have it so that 1 DNA molecule consists of both of the old strands, and the other consists of both of the new strands, but that's not how it works in nature. The 2 DNA molecules consist of 1 old strand and 1 new strand, and the same thing on the other DNA molecule. So replication begins at an origin of replication. This is where in eukaryotic cells like ours, there are multiple origins of replication because our DNA, there's so much of it, you can't just start at one place. It would take forever to replicate it. So our eukaryotes have multiple origins of replication. With prokaryotes, their DNA is much smaller, so you can have 1 origin of replication. So here's our RNA primer, and our DNA is being synthesized. It's being synthesized in the 3' direction. Whenever you make a new strand of nucleotide, it's always synthesized in the 5' to 3' direction. In other words, a new nucleotide is added to the 3' end. Okay? This is problematic for one of the strands, because you can't synthesize it in the, once you reach the end of the replication bubble, you can't keep going forward, you have to go backward and synthesize it in the other direction. So that's, the strand that's synthesized continuously is called the leading strand. The strand that has to be synthesized discontinuously is called the lagging strand. And this lagging strand creates a whole cluster of what are called Okazaki fragments. These have RNA primers all throughout the strand. These are sealed finally by an enzyme that's called DNA ligase. Yeah, so helicase unzips the DNA strands. The leading strand is synthesized continuously. The lagging strand is synthesized discontinuously. DNA polymerase can only add nucleotides to the 3' end. This is difficult to see to understand sort of without a visual. But I think for our purposes, you don't need to really get that deep into it. You just need to know that there's a leading strand, there's a lagging strand. The leading strand is synthesized continuously. The lagging strand is synthesized discontinuously.
When we started playing technology, can you just break down what the discontinuously means again?
It means you can't synthesize it continuously. You can't keep synthesizing it in one direction. You have to synthesize in one direction, stop, go back behind it, synthesize it, go back behind it, synthesize it. So it's much slower, and because you have to lay down an RNA primer each time, that produces what are called Okazaki fragments. So here you have these single strand binding proteins. You have the leading strand, you have one RNA primer, and it's being synthesized continuously. The lagging strand, you have multiple RNA primers, and it's being synthesized discontinuously. I think that was it already. So this is how the process works in bacteria. I'm wondering, what is this? This is DNA replication. So you need to initiate the process of DNA replication. This is called initiation. This involves DNA helicase unzipping the two strands, etc. Once you reach the point where DNA polymerase elongates the RNA primer, that's called elongation. And the point at which you end the process is called termination. Something like a video. I don't know how to play it.
You have to enable it in this thing.
Was it start of process and what was the middle one? Did you get it?
This one? Their lungs, their hearts, their kidneys, and then you could take organs from the pigs because they've been edited, because they've been engineered to make them